So it is a pleasure for me to introduce Dr. Zachary Smith. Zach has graduated in 2009 from the University of Rochester with a degree in optics. He then moved to the University of California, Davis, where he stayed for 6 years, going from a postdoctoral position to a project scientist position and then NSF entrepreneurial fellowship until 2015. And in 2015, he moved to China. Very interesting move. He moved there at the University of Science and Technology of China in the Department of Precision Machinery and Precision Instrumentation as a professor. And I was part of the 1,000 Young Talent Global Recruitment Program that China has in place. And he has been there for 3 years. He's working on optics-based point-of-care diagnostic techniques. He will share with us some of his newest results.
Okay, thank you. Can everybody hear me okay? Okay, so thank you, Sebastian, for such a nice introduction. So I'm from the University of Science and Technology of China, from the Department of Precision Machinery and Precision Instrumentation. If you're interested, I'm going to talk about the work that we do on point-of-care diagnostics. We also have some research efforts in super-resolution microscopy and also Raman spectroscopy. If you're interested in some of the other things that we do, please feel free to visit our website. So I also want to acknowledge up front all the many people who I work with during all of this experiment. So I'm from a very large multi-investigator laboratory. So we have 6 or 7 faculty. In particular, here's Kai-Chin Chu, my wife, who's extremely big part of all of this work. So she's basically my collaborator on all of this stuff, as well as all of my excellent PhD and master students. Our collaborators, including Professor Waxman, and as well as our funding agencies. Let's see. So many people here may not be familiar with USTC. So USTC is located in Hefei, which is the capital of Anhui Province. It's a small city with a population of 8 million people. And it's relatively close to Shanghai. This dot here is Nanjing, so these cities you might have heard of before. And USTC is a very important school in Chinese history. It's one of the first schools started after the beginning of New China. And we're here on the west campus of USTC. So this is our engineering school, and this is Pretty Lake on campus. So it's not Hefei is a rather industrial city, but USTC is a nice-looking place. Anyone here is welcome to visit anytime. And then when for the talk today, where we're really starting from is that we all have this dream that we want to have equal access to high-quality healthcare. No matter where you live in the world, right? We all hope that, 100 years in the future, if you get sick, someone's just going to come and wave a little magic wand over you, and then you're just going to instantly tell you what's wrong with you. So the reality is that if you want to know what's wrong with you, you have to go to the hospital, right? So the hospital has these very big centralized equipment, centralized facility, and these diagnostic instruments, they're very powerful, and they're very good at what they do. So you can get a very accurate, very high-quality diagnosis today, but the problem is that you have to go to the hospital to get it. Because these instruments, they require highly trained technicians to run. They're very expensive. They require very large power sources. And so, if you live in a part of the world where it might take you, several days walking to get to the hospital, right? That's a huge burden on you to get healthcare, right? So if you aren't feeling well, you have to weigh, well, what's the economic cost to me to leave work for 3 days versus the health benefit or health consequence of leaving something untreated. So, we've been working on this problem together for a long time, right? So this is work that we did back at UC Davis back in 2009 or 2010, probably we started, and then continuing on for a few years. And we made some interesting things using cell phones, right? So we made a cell phone microscope, a cell phone spectrometer, a cell phone particle sizer, all of these different instruments that we felt had utility in a clinical setting, right? So you can bring this phone with you anywhere in the world. You can take images of malaria, of plant parasites, of all of these different things, and maybe you can use that for some kind of low-cost diagnostic. So the question that we have to consider is how easy are these instruments to use, right? So for example, what we found is that the performance can be highly dependent on the user. So here's the instrument that we put together in our lab. So not that I'm so great, but that I have a optics training, so I know how to put a microscope together, so that I can, in my hands, this microscope can see 1 or 2 micron resolution, which is high enough to see malaria parasites. But this same exact microscope in the hands of somebody else, so this is work that we weren't involved in, where there's a medical technician who's putting these things together, and not that necessarily that they're not good, but they just don't have an optics background, right? Their training is in medicine. And so when they make a cell phone microscope, the images don't have good quality, and even very large parasites can't clearly be imaged. So therefore, when you think about what's the diagnostic sensitivity or the diagnostic accuracy of such an instrument, unsurprisingly, it's not very good, right? It's very little better than chance. So that if we want to have medical systems that are designed to be operated by untrained users, some kind of handheld instrument may not be the best choice, right? So,
sorry,
yes?
Is the difference we're seeing here difference between the instrument or between the user or both?
I would speculate, so we weren't involved in this work, so I can't answer that question exactly, but I would speculate that it's both. I would speculate that there's probably some sort of motion artifact because maybe the person is not holding the sample in perfect focus. And then additionally, the lens, there seems to be some kind of doubling or something of the image, which may be caused by some sort of strange illumination or something like that. So, but there was a reason why we chose to use cell phones in the first place, right? So they're very cheap, but they're very powerful because they have a lot of very excellent commoditized technology that's been put inside of them. Things like LEDs, cell phones, cameras. They have an onboard computer, wireless communication, all of these different things. And so the idea is that maybe we can take all of those individual technologies and put them together in a standalone package that maybe could even potentially be less expensive than the cell phone, although also potentially less general. But maybe that kind of off the cell phone design will allow you to make different design decisions and make maybe different engineering decisions that will make the system ultimately have a higher diagnostic performance. So this is just a very simple example. So this is an automated microscope that we built, and the guts of this microscope are not that dissimilar from the cell phone. So here we have a camera lens, which is imaging onto this low-cost sensor. So this is pretty much exactly what you would have inside of your cell phone. And then we add a second lens to it, just like we did with our cell phone microscope. But now this thing is on a stabilized stage. And then we hook this stage up to motors that we control with a Raspberry Pi or Arduino. And this system, because we add these automation components, now this system can have auto-focusing, auto-scanning, all of these different capabilities while not being any more expensive than what you would pay for a cell phone, right? So one of the things that we did when we designed this system was that we designed it to use only off-the-shelf components. So nothing that's custom made, nothing that's custom designed. So the idea is that ultimately you can put something together which is as easy to put together as a Lego set, or maybe slightly more frustrating than a Lego set, but maybe like a piece of IKEA furniture or something, where as long as you have the instructions, it's really not that hard to put it together. And then in addition to being easy to assemble, it also has these fancy capabilities, right? It can take bright field fluorescence. It can auto-scan, auto-stitch. The scanning has better than 1 micron positioning accuracy. The resolution is 1 micron. The field of view is greater than 1 square centimeter, while still being only about $300. Right, so that way that we can just say, well, all of these things can just basically slot together one on top of another, so that ultimately you don't need anything more complicated than a hex wrench to put this together. So then if we go back to these parasite eggs, so this is what is able to be achieved by a cell phone microscope that's being hand operated. And then our system, you can see that our image quality is much higher, and it's also much more repeatable. So that when we have these fully automated systems, then maybe we can actually use this to actually do some parasite diagnosis in the field. So in China, the veterinary situation is a little bit different than in the US. Most of the animals that are raised on small farms and small shareholder farms. So there might be somebody who's raising, somewhere between 1 and 20 goats and not, kind of this huge sort of factory farm infrastructure. So that means that if the animal is sick, it's actually a little bit more challenging because there might not be a veterinarian on site who's going to be able to deal with this infection. Which leads to overtreatment and other things. So, these goats, they pretty much, as I'm sure everybody knows, goats are famous for just eating anything they come in contact with. And there are a whole bunch of parasites who they have evolved to take advantage of that kind of behavior, right? So they're hanging out on leaves or in droplets of water on grass, and they're just waiting for the goat to come along and eat it, and then the goat is going to become infected. So, how do we deal with this? And then anybody who knows me well knows that I'm a big cat person. So, in fact, this also hits close to home. One of my cats, he used to go outside. He doesn't go outside anymore. But he ate an infected mouse and got infected with a tapeworm. So, the classic diagnostic method to diagnose these parasites is to collect some scat from the animal and then mix it with a flotation solution. And so this flotation solution is nothing more than sugar water. And so this has a very specific density, so that if I then mix the poop together with the flotation solution, I strain it to remove any large particles, and then I put it into a slide like this, which is called a McMaster slide. And basically most of the debris in the sample floats to the bottom, and then the eggs and some other debris rise to the top. So that then I put this onto a microscope, and this microscope doesn't have to be anything fancy, right? So this can be done in the field, and then I image these eggs. But the problem is that I have to, in order to get good diagnostic accuracy, I have to scan over a very large field of view. So I have to scan this entire square in order to get an accurate clinical count. So, and then these images, they look like this ultimately. So this is after all of the straining and the flotation. So we've done a lot to remove a lot of the debris from this sample, but it's still very, very dirty. So that means that I have to be relatively highly trained to look at this image and understand what's going on, right? So I'm not sure that my student went through a training to do this. I haven't. So, to me, all, like maybe these three things, they all look sort of the same. Maybe this guy, they all look roughly egg-like. But only one of these things is a real egg. The rest of them are all just debris. So what we thought is that maybe we can use our automated microscope. This is exactly the same as the microscope I showed you before. It just looks nicer because we added some 3D printing around it to sort of make it a little bit more stable. And we can image over 1 square centimeter. So we can image this entire McMaster chamber. And then the image has 1 micron resolution so that we can image these eggs with high precision. And then this is a very challenging diagnostic requirement because if there's any more than 3 eggs in this entire chamber, then the animal must be given some kind of treatment. If there's less than 3 eggs, the infection might be transient or whatever, so that usually they recommend just a follow-up. So we can image this, and we could imagine taking this digital data and transmitting it to a trained user someplace far away. But another thing that we could imagine doing is trying to make the diagnosis automated. So the problem is that this is a very difficult image processing problem because this thing is very dirty. There's lots of objects in this image. So how do we separate these objects from the background? So very luckily, one of my colleagues, Ming Zai, he's an expert in deep learning, so that we can then train a deep learning network to identify where in these images are the eggs and where in the images are debris, and do this kind of automated segmentation. So once we do that, so here's just some details about this training. So we have these manual labels and training data. These two things are combined together in the network to produce a trained network. Then new unseen data is fed into the network, and the segmentations come out. And then we do a small amount of post-processing on the segmented images, and then we get a diagnosis and an egg count. So the egg count will allow us to monitor the severity of the infection, whether the animal needs treatment, whether the animal is responding to treatment. And we can see that our hand counting versus automated counting is rather accurate. And then it's even accurate even at this low end of the curve, where that diagnostic decision is being made. So even when there's small numbers of eggs, we can identify those eggs relatively accurately. So our diagnostic precision is about 96%. So we give the right answer, give medicine or don't give medicine, 96% of the time, even when we're having something where the imaging is fully automated and the analysis is fully automated. Then over here, we can look at the morphology of these different parasites in order to determine what kind of treatment is required, right? Because all of these different parasites, they have their eggs have different characteristic morphologies. We can extract that and be able to diagnose, okay, this parasite below this boundary is a protozoan parasite. These ones up here are like worms, nematodes, and so they get different classes of drugs. Then even among these classes, right, so Trichuris or Ascarid, they might need different kinds of drugs, or different, some might need more aggressive therapies or not, and we could even make decision boundaries here if we needed to. Because this is very simple, now we don't need to use the veterinarian's valuable time, right? The veterinarians, they don't particularly like to run these assays because it's very laborious for them. It takes a lot of time. It's very tiring to look through these whole fields and try to count the number of eggs. So that generally leads to overtreatment, and this overtreatment has led to drug-resistant populations. So this is an example of a goat whose treatment we monitored over time. So at day -5, that's when the infection was diagnosed. Then 5 days later, the farmer was given the drug, and he gave it to the animal. So at day 0, that's when the treatment started. And then we can see that over the first several days, the animal is the parasite population is dropping very dramatically. But after about a week, that parasite drop-off stops. And then the parasites start to recover, because there's some subpopulation within that group who are resistant to the drug. So that means that not only can we diagnose the animal, but we can follow its response to treatment, and then maybe we can advise that either a second dose of the drug should be given, or a second-line therapy should be tried. So now I'm going to switch to a slightly different topic, which is blood counting. So this is another place where there's a need for low-cost or portable diagnostics, right? So the complete blood count is something that's one of the most commonly ordered diagnostic tests. So anytime you go into the hospital and you have blood drawn from your arm, they're likely going to run a blood count, because this kind of gives an overall window of your health, right? So they're going to count, what are your red blood cells? How many platelets do you have? What are your white blood cell counts? What kinds of white blood cells do you have? What's your hemoglobin content? So these things can be used to diagnose anemia. They can be used to monitor infections. They can be used to monitor the response to chemotherapy. They can be used to look for signs of internal bleeding. And so it's a common starting point for many diagnostics. So if we could take this kind of blood counting technology, which is only available currently in the clinical laboratory, and move it outside of the clinical laboratory, then some of these screening tests and other, sort of first-line testing could be done at a local clinic, and someone could be advised, you probably just have iron deficiency, why don't you take an iron tablet and go home? Or, say, well, looks like something is going on, and maybe, you should go to the hospital. Kind of do some sort of first-line triage. So we were funded at the beginning of this work by the Tahoe Institute for Rural Health Research. So their interest is bringing healthcare to rural populations, right? So this is not just in low resource settings, but also in other settings where access to healthcare is limited. So up in Tahoe in Northern California, people live up in the mountains where there could be, many feet of snow, and it could be a big burden for them to come all the way down the mountain to Sacramento, where we were starting this work, to actually get a test run. Another thing is that, again, this also has applications in animal health, right? So this is another one of my cats. I have 2. So this is Sprout. And he recently had feline diabetes. So diabetes is interesting in cats. They can have it spontaneously and recover from it. So he had a bout of feline diabetes. So during that time, he was constantly having his blood tested. And any cat owner who has been through the process of having blood drawn from a cat, it's very unpleasant for everybody involved. The vet doesn't like it, the cat doesn't like it, the owner doesn't like it. And so if we can find some way to make that sort of testing easier or more pleasant, that would be beneficial. So the typical complete blood count is measured with this big system like this, right? So this is a complete automated hematology analyzer. It's basically a very specialized flow cytometer. And so this instrument has tons of different reagent paths. The blood is taken automatically from a tube and then split into different paths. Reagents are automatically added. The cells are passed one at a time through multiple detection channels. And then they pull out all of this diagnostic information. So this instrument is really beautiful. The problem is it's very large. And it's also very expensive. It requires very routine calibration and maintenance. And it's not suited for places outside of a clinical laboratory. So we have another system which is a small, simple to operate, self-contained microscope, kind of similar to the automated microscope we showed earlier in the talk, but just with some additional specialized components for the blood counting. So for example, for the blood counting, we take multiple colors of fluorescence. So we add in a little inexpensive filter wheel. And the other key piece of this is the simple sample preparation. So typically, in order to get your blood drawn for a complete blood count, you have to have your blood drawn by a phlebotomist, right? So this is the person who's trying to find the vein in your arm to get to poke the needle into to draw a few mils of blood. And some phlebotomists are better than others, right? And sometimes they miss. Oh, sorry, I have to poke you again, right? And so you can imagine doing that on a cat where their fur is covering where their veins are or whatever. It's a very painful process. So one of the interesting things about our technology is that rather than requiring 100 microliters or several hundred microliters of blood, ours requires actually less than 1 microliter. Here we draw 10 microliters, and we draw it into a capillary tube. So this is compatible with a finger stick or maybe a paw stick for an animal. The blood is drawn up into a capillary tube, which has a precise length so that the volume can be very precisely controlled. Then it's dropped into a tube of pre-prepared reagents and shaken up, and then put onto this chamber. And after that, everything is automated. So the sample imaging and the sample analysis is fully automated. And because it requires a very small volume of blood, this is good for animals. It's also good for babies. So oftentimes, infant blood is drawn using a heel stick. So you can imagine trying to push a milliliter of blood out of a baby's heel. The parent is probably not going to be very happy when this blood draw is happening. So one of the key things about this is, sort of the magic of what's happening inside of this reagent mixture. So this is work that was done in collaboration with my colleague Tingjuan Gao, who's an analytical chemist. So she was responsible for a lot of the reagent design and reagent preparation here. So when you think about imaging blood, one of the things is that blood is a very complicated soup of stuff. And there's lots of different kinds of cells. The cells have all sorts of different shapes. And we try to find some way to pull all of this information out in a very easy way. So if you just take blood, dilute it, and put it onto a microscope slide, you get an image that looks like this, where because the red blood cells, which are the most dominant form of cells in blood, they're kind of pancake shaped. So they have all these different orientations. When they fall to the surface of the chamber, they could fall on their edge. They can stack like a little pancake stack. And because they're not round, they kind of have this strange aspect ratio. They can tend to kind of surf on currents and float for a long time. So it's very challenging to get a count of the red blood cells from an image like this. So what we do is we play a trick that's common in the flow cytometry community, where we add a small amount of surfactant to the blood, and that causes the blood to pop from these pancake shapes onto a sphere. So now when things fall in a sphere, there's no orientation. If one cell falls on top of another, it rolls off, so that you can get these very, very uniform images, these very uniform kind of sparsely packed monolayers of blood. And because all of these red blood cells look the same, it's very easy for us to take image processing methods that are 60 years old and apply them very easily to something like this. So we don't need to train a deep learning network. We don't need to have a huge amount of processing power. We can do this very, very easily, which means that ultimately if we want to take this instrument and package it to be run on a Raspberry Pi or some other very inexpensive computer that has the computational oomph to deal with this kind of data. So we can get about close to 100% accuracy in counting these cells. We also add a small amount of fluorescent dye. This fluorescent dye is acridine orange. And acridine orange is a really beautiful dye for looking at white blood cells because it doesn't fluoresce very strongly until it binds to something. And then depending on what it binds to, its fluorescence shifts. So it has both a monomeric form where it fluoresces green and a polymeric form where it fluoresces red. And so that means that we can use not only the staining, but the color of the staining to pull apart where are the white blood cells and what kind of white blood cells do you have, so that we can use the color to pull apart a three-part differential. So we can tell, how many lymphocytes, how many monocytes, how many granulocytes do you have. And again, we can use very, very simple image processing methods to do this so that this kind of analysis can be run using relatively low computational power. Acridine orange also stains platelets. So these little tiny guys are the platelets. The big ones are the white blood cells. And when we take an image like this, because we're not using some kind of $80,000 scientific camera, there's a relatively large amount of noise. And the platelets themselves, their fluorescence is relatively dim. So some of them are really right at the noise floor. The signal to noise ratio is only slightly better than 1. Very luckily, my wife is an expert in image denoising. So basically, we can use her sort of secret sauce called total variation constrained denoising. And we can take an image that looks like this. I hope you guys can see that there's sort of a little bit of salt and pepper fuzz to this image. And after the denoising, that just magically disappears. And it disappears in a way that preserves the structure of the sample so that all of the platelets are still, their intensity is preserved. So that actually before and after this counting, before and after this denoising, the counting becomes really accurate. Additionally, when we run this test, we've run tests with our instrument in both a laboratory setting as well as a field setting, run by highly trained users, which is basically me and my students, the people who built the system and know exactly how it works. We have people who are minimally trained users, so those are people who we've described the system to them, shown them a protocol, demonstrated to them how it works. And then we have people who are totally untrained, people who we basically put them in a room by themselves with the instrument. They get a protocol that describes to them how it works, but no one has demonstrated it to them. And we look at how well these different users can extract the blood counts from this kind of system. And what we find is that there's basically no statistical difference between the most trained and least trained user when operating this kind of instrument, because we've done all of this effort to try to automate the image acquisition as well as automating the image analysis. So interestingly, even in a clinical laboratory, there's a need for this kind of device because our blood counting method doesn't rely on flow. It relies on this kind of large field of view imaging. So that means that in situations with a flow cytometer where once the cell count drops below a certain value, the flow cytometer is no longer accurate. So for body fluids that are very acellular, like cerebrospinal fluid or synovial fluid, that the majority of clinical samples can't be counted automatically. So in a hospital like the UC Davis Hospital, they might get 100 such body fluid samples a day, and there's someone whose job it is to just spend all day looking through the microscope and counting these samples by hand. So in fact, our system can accurately count these samples with a limit of detection basically 100 to 1,000 times lower than commercial clinical systems. And then it doesn't just work on people, but it also works on animals. So we've tested this on humans, horses, cats, dogs, cows. We've even tested some yaks. So that means that now because we have all of these nice things, the instrument is small, it doesn't require a highly trained user, it doesn't require a large sample volume. Now you can imagine that this thing becomes more general, right? So for example, if any people here are doing mouse biology research, right, it's very unusual to run a complete blood count on a mouse, even though you might be interested in its sort of overall health state. But the problem is that the current clinical instrument, if it requires 100 microliters of blood to work, by the time you get the blood out, the mouse is dead, right? It only has 100 microliters of blood. So now we can take, well, maybe still a rather large percentage of its blood, but still enough that it can survive afterwards, and maybe do more routine health or sort of overall testing of the mouse's immune system. So the last 5 or 10 minutes, I just want to describe to you guys about some work we're doing to diagnose anemia. So anemia is a very widespread public health problem. It's probably the most common, I guess it's probably the most common disease in the world. I think a third of the world's population is anemic. And you can see that this is a WHO chart which plots the severity of anemia in different countries in the world. And you can see that the places where anemia is really in a critical situation are exactly those places in the world that have the least access to high quality diagnostics, right? So it's very hard for someone to find out whether they have anemia, and if they have anemia, what kind they have. Because one of the interesting things is that anemia has many different forms, right? So the most common form is iron deficiency, right? So you didn't have enough iron in your diet, and so therefore you can't produce enough hemoglobin, so that you can't produce, so that you then become anemic. So if that person is has iron deficient, then the solution to that is just to give someone an iron tablet. And it's very simple to do. Iron tablets are very cheap. So you might think, well, why don't we just supplement everybody with iron, right? Just like many places in the world supplement the population with fluoride or something else. So there was actually a study done several years ago where they tried this kind of broad population, broad spectrum iron supplementation. And they found that giving somebody iron who doesn't need it can be damaging to their health, because certain groups of people have a reduced capacity to process iron. Iron, if you take too much of it, it can become toxic, so that they're more susceptible to what's called toxic iron overload. The other thing is that one of the reasons why genetic anemias persist in our population is because they confer an advantage against malaria. So the malaria parasite needs to dispose of the byproduct of the hemoglobin consumption. And if you have a genetically altered form of hemoglobin, it can't dispose of that byproduct in a safe way. So people who are heterozygous for sickle cell anemia or thalassemia trait, they have an evolutionary advantage against malaria. If you give someone iron, you actually tend to remove some of that advantage, so that people can have these negative health consequences by being supplemented with iron when they didn't need it. So what we need to do is find a method which can both diagnose anemia, as well as diagnose what form of anemia you have, so that you can accurately guide treatment. So even in China, which is a relatively well-developed country with a well-developed clinical ecosystem, iron deficiency is still persistent, right? So 10 to 20% of Chinese children suffer from iron deficiency. And in Southeast China, about 10 to 20% of the population carry thalassemia trait. So that means that if someone comes into your office with something like an anemia presentation, you must first test to figure out what form of anemia do they have. Again, if you have iron deficiency, iron is required. If you have thalassemia trait, iron is dangerous. This is that study I alluded to earlier in The Lancet, where they showed that giving someone an iron tablet can be very detrimental to their health. But yet, if you leave iron deficiency untreated, or if you leave anemia untreated, it can have these outcomes among vulnerable populations. So in children, you can have slower cognitive development. Among pregnant women, the pregnancy can be more dangerous. And among the elderly, there's a reduced immune system and increased susceptibility to infection. So the current clinical workflow might look something like this, right? So we have a patient who comes in, they have a complete blood count run, which tells them what the hemoglobin value is. And if it's normal, you would assume that that person is healthy. If it's abnormal, you would say, well, then they need to have some kind of iron test, right? So if that iron test is low, they're suspected to be iron deficient. If it's normal, then they might have confirmatory tests done by PCR or gel electrophoresis to determine what kind of genetic hemoglobinopathy do they have. So when you imagine the initial patient coming in, 75% of the population at least probably doesn't need to have any tests done, right? They're already healthy. So there's a whole bunch of tests that could be run that are not really necessary. So if we can find some way to take some of this information and bring it outside of the clinic, then we can save these very expensive and laborious tests only for those who need it, right? So if we can take some kind of, make some sort of anemia screening tool, then we can test everybody in the whole population without requiring that the whole population eventually go to the hospital. So people have known for a long time that, that these different forms of anemia lead to different changes in the red blood cell morphology, right? The classic example is sickle cell anemia, right? Where the red blood cells have this sickle shape. So thalassemia trait is no exception. So thalassemia trait tends to have, patients with that tend to have very small red blood cells. People who have iron deficiency have slightly larger but still small cells, and then healthy cells are up here kind of at the normal value. So the problem is that these differences are very tiny, right? If we imagine going from this volume down to a radius, these different things have only 100 nanometers difference in their radius. So that's well below microscopy's resolution limit. So it could be very challenging to be able to view that using, let's say, one of these low-cost microscopes that we've been talking about earlier in the lecture. So currently, the red blood cell shape and size is again measured using this automated hematology analyzer, where they use light scattering, or they use the Coulter method, where cells are passed through a charge aperture, and they monitor the resistance change as the cell passes through that aperture. So this is how these things are typically determined. But again, these instruments are big, they're bulky, they require a trained operator. So we know a little bit about elastic light scattering because we have this training in optical physics, so that we can use elastic light scattering to also get this kind of nanoscale sizing information. So here I'm showing you a plot of the intensity of light scattered by a particle into different angles. And you can see that there's sort of this fringe pattern where there's peaks and valleys in that scattering. And then the pattern of those peaks and valleys changes very rapidly as the particle grows in size. So this scattering intensity versus angle is a highly sensitive measure of particle size. So we showed back in 2012 in work I did with Professor Waxman, where we can show that if you shine a laser onto a sample with some very, very simple optics and a cell phone, you can actually get you can actually measure these intensities of scattered light versus angle. So you can imagine going, let's say from here, which is 0 degrees, as I go out here, this is increasing angle, and you see there's these peaks and valleys in the scattering pattern. And if we analyze that with me theory, we can extract particle sizes whose mean size is accurate to within 15 nanometers, even though we're using these very, very simple consumer electronics. So here's sort of the second generation of that system, which looks more complicated. But it's actually not very complicated. So the key thing is that we add a second laser, and then instead of bringing the laser in right at 0 degrees, we bring it in from an oblique angle. So this allows us to not measure the center to center out and get this kind of angular redundancy, but we image this sort of slice of the angular scattering space. So this allows us to get an increased number of scattering angles. And we are very lucky that we have a very good clinical collaborator in China. So my collaborator, Dohu, so he's an investigator in the Children's Hospital of Chongqing Medical University. So this hospital sees thousands and thousands of patients a day. And he is saying that anything that they can do to get the test not run at the hospital is a benefit to them. Because in China, you probably know that the majority of families only have one child. So that means that if there's a child who's sick and they have to go to the hospital, it's not just the patient who's coming, but it's the patient, it's mom, dad, aunt, uncle, grandpa, grandma, the other set of grandparents, right? So for any one patient who comes in, they have to have space for 10 people to be there. So if they can move that outside of the clinical laboratory, that's a benefit to them. So he's very, very generous with his time and very generous with his space. So he allowed us to actually take our instrument, which is built in Hefei, transport it to Chongqing, and actually sit inside of his clinical laboratory and run samples. So what we do is we collect these scattering patterns. We do some very simple image processing to extract these one-dimensional curves, basically an angular spectrum, right? So we get the intensity versus angle. And we can do some calibration here. We're showing how we calibrate using some NIST traceable particle sizes. But one of the reasons I wanted to show these plots is that you can see this is the theoretical scattering, this is the experimental scattering, and these things agree with each other really, really well. So this means that the instrument we're making actually can be analyzed very well using just some fundamental physics. We play this exact same trick as we played before of sphering the red blood cells. And we see that when we do that, now the scattering from the red blood cells looks very similar to the scattering of these NIST traceable particle size standards, right? So we get these very nice fringes that we can use to analyze the cell size and shape. When we do that, we can get the mean cell volume, the mean cell hemoglobin concentration, which is related to the refractive index of the red blood cells, and we get the red cell distribution width, which is how many different sizes of red blood cells does somebody have. And the agreement is not perfect, but the agreement is pretty good. For some of the parameters, we get almost as good as the clinical system. And you can see that there's actually quite a strong separation between healthy iron deficiency and thalassemia trait, especially on this mean cell volume parameter. And because we have our generous clinical collaborator, within just a few weeks, we're able to run a pilot clinical trial where we can look at blood from 250 Chinese children, among whom many of them have iron deficiency and thalassemia trait. So we can take these three parameters and doing some very simple machine learning can come up with some decision surfaces so that a new sample that's brought in can be diagnosed as anemia or not. Among the people who are anemic, do they have iron deficiency or thalassemia trait? So we can diagnose anemia versus healthy with about 100% accuracy. And iron deficiency versus thalassemia trait, it's a harder problem to crack. But right now we have about 90% accuracy. And again, our goal is not to reach a final diagnosis. Our goal is really to try to identify who should go to the hospital and who shouldn't. So we think even with only 90% accuracy, this is still a good number for a screening test, right? So that basically we can err on the side of caution, send a few extra people to the hospital, so ultimately everybody gets the right diagnosis, but the number of resources utilized to arrive at that diagnosis is minimized. So what causes our misdiagnosis? So basically the most common problem we have is we have people who are iron deficient who we claim to have thalassemia. And so basically, among iron deficient people, there's a loose correlation between how iron deficient you are and your cell size. So if you just have a little bit lack of iron, your cells are going to look normal. If you have a severe lack of iron, you've been iron deficient for a long time, then your cells are going to get very, very small. So really, it's this group of people who we're worried about. And the thing is that if we say anyone who presents with thalassemia trait should go to the hospital to have a genetic test done, well, if these are the people who we're sending to the hospital, they have severe anemia to begin with, so maybe it's not such a bad idea they go see the doctor. And we started all of this work just trying to do some very low cost sort of frugal science and try to be able to use this outside of the laboratory. But some of these ideas have applications inside the laboratory too. So this is some work that I was involved in where here we have exactly the same optical system. So we have a cuvette, we have a laser coming in, we're imaging the intensity of light versus angle. But the difference is that instead of using a $5 DVD player laser and little low-cost camera, we swap the camera out for a very expensive CCD. We swap the laser out for a $500,000 pulsed laser system, and all of a sudden we can actually do some interesting photochemistry where we can look at the nonlinear properties of metal organic frameworks as they're growing in solution with very, very high time resolution. So quickly to summarize, what we're trying to talk about here are diagnostics that can be used anywhere by anyone. So when you think about that, then you'd have to think about not just the instrument itself, but also how is that instrument going to be used? Who is the user, right? Everything can't be operated by a PhD in the laboratory, right? You have to think about some of the automation, and both in terms of the instrument as well as the analysis, in order to get some kind of reproducible or actionable clinical information when you transfer it outside of the laboratory. And then we showed a few different examples here where we have blood and other body fluids where we can get diagnostic information essentially equivalent to clinical gold standards. For example, parasite diagnosis, complete blood count, or anemia screening. So with that, I'll wrap up and welcome anybody's questions. So thank you very much.
Thank you. Questions?
Thank you. Oh, thank you. It was fantastic. Just for the first topic, because even now, the people should wash their samples perfectly and minimize the amount of dust in it. So why you can't use any fluorescent antibodies for the parasite or something to make it easier even to visualize it even when it's not perfectly clean?
So this is a really good question. So there are people who are working on that. So one problem is that when you add the fluorescence, right, that's a sample preparation and sample storage, which is very difficult, right? So these antibody things oftentimes have to be stored carefully. But another thing is that these parasites, they're just the worst, right? So they've evolved very carefully to evade the host immune system, the digestion process, all of these different things. So one of the ways they do that is that when they're in their kind of ovum or egg state, they have their egg, which is growing, which is surrounded by a shell, which is surrounded by another shell, which is surrounded by another shell. And it's very hard to get things inside where they can actually be stained. So there is a group that showed a very nice paper where they can image these parasites using a smartphone with fluorescence. But in order to do that, they have to boil the samples to peel away these protective layers and allow the sample to be stained.
Questions?
You talked about using deep learning to train your classifier. One question was how many manual classifications did you have to do? Typically, deep learning requires a lot of...
It does. So we had, I think, on the scale of 500 manually annotated examples of eggs, and another scale of 500 of images we took from healthy samples where we're trying to identify the debris. So in those, the manual annotation is easy because there's just no annotations in the image. But we do have to do these manual annotations of something like 500 to 800 images of eggs, which oftentimes have many eggs inside of them. So it is a big burden on the upfront work to do that annotation. The hope is that once you train that network, that work only has to be done a single time.
You mentioned, on the slide, it mentioned support vector machines, and it wasn't clear to me. When you say deep learning, you're talking about neural network?
Yeah, so I skipped over that, but let me go back to it. So basically what we do is, because these samples are so dirty, even if you have, let's say, so our deep learning network is something like 99% accurate, but even if it's 99% accurate, there's still going to be a few false positives in the image because it's such a dirty image. So what we do is we pass it through the deep learning network, which is like this kind of UNet structure, and then afterwards, we get this segmented image. The segmented image has the eggs as well as things that the deep learning network identified as looking similar to eggs. But what the deep learning messes up on, those things are morphologically dissimilar from the real egg. So then what we do is we extract kind of like the eccentricity and area and other things about these objects, and then use a support vector machine network to separate the false positives from true positives.
Hello. On? Yeah. Okay, thanks a lot for the nice presentation. I was really impressive, all the things you have accomplished. So maybe the one thing I want to first to ask is about the, you said you're 1,000 times more sensitive than the clinical diagnostics, but so why can't they make their instruments more sensitive? Or why is...
I don't know the answer to that question. I guess you'd have to ask those companies, but I guess basically those automated hematology analyzers, they're very specially designed to work on blood, right? So blood is very extremely cellular. So they have these very high dilution factors from the sample to the measurement side. So when you try to do that with a sample that you're going from something that has 1 million cells per microliter, something that has 10 cells per microliter or 1, right? Then, even if you don't dilute at all, now your counts and other things are going to be inaccurate just because your flow speed and other things just can't be controlled that accurately. So I guess maybe the answer is that flow cytometry just has a hard time to work with these very, very rare samples, right? That's not really what it's designed to do.
Yeah, let me another question. So I mean, for global health, one of the criteria usually uses is assured criteria, which is affordable, sensitive, specific, and one of them is kind of instrument-free. So, I mean, I think that's maybe hard to realize, but how? So, because you spoke about using this in really low-cost regions, but there's still kind of an instrumental setup here,
That's right.
and maybe needs electrical connection.
Exactly.
So
Right.
what are the obstacles, and how can you envision going a step further and removing even more of your instrumentation there?
So that's a tough question, right? So that's tough to solve. I think the more you try to reduce the instrumentation, right, then you get to things like dipstick tests and other things which don't require any power. I think that's a very difficult question to solve. There are people who are doing beautiful, beautiful work on that. There was a paper recently in Nature Biomedical Engineering where they developed a thing called a paperfuge, and it's this hand-powered centrifuge which can operate at 30,000 Gs, several 100,000 RPM, and they can use, if you imagine, it's this disc that spins as you pull, kind of like a toy, and you can imagine 3D printing some sort of microfluidic circuit onto that thing and doing some kind of sample pre-processing. So I think there is a lot of excellent work being done there. For our stuff, I guess probably what I would say is that our instruments are designed to go maybe someplace slightly between the lowest tech environment and the sort of standard centralized medical infrastructure. And I guess maybe I would hope that, as our stuff starts to become more mature, hopefully the poverty in the world is also being eased, and these places are becoming more stably electrified and things like that. But again, it's a very challenging problem.
Thanks.
Yep.
Okay, thank you for the presentation. Regarding what you mentioned about testing small samples of blood, so I was reminded of 2 years ago, there was that company called Theranos.
Mm-hmm.
They built $9 billion company based on false claims.
Yep.
So what are ultimately the limitations of the testing small samples, and what information can you get and not get from them? And in the long term, can some of these limitations be overcome by new technologies like, say, deep learning?
Let's see. So it's an extremely good question. I think one of the things that Theranos is trying to deal with is that they're looking at these kind of biomarkers and other things that have relatively low concentrations, where they're using standard instrumentation which isn't designed to be used on small volumes of blood, right? So for example, if you have we're talking about these automated hematology analyzers and how sometimes they don't deal with small numbers of cells. They become less accurate as the cell number goes down. So in that system, if you take 10 microliters of blood and you just dilute it 10 times and you feed it into the instrument, then you're dealing with something that's 10 times less cells than the instrument was designed to handle. So I think part of the problem with Theranos is that I don't know all of the details. My understanding of that issue is that they are basically using standard laboratory instrumentation. Yeah, they're out of business, but they were using standard laboratory instrumentation just totally outside of the quality control validation cycle for those instruments. So here I think maybe some of the stuff that we're doing is trying to say, well, we're starting with a small volume of blood, right? And how can we make the best use of that volume of blood, because we know that we don't have 100 microliters of blood to spare. And so I think there are different engineering choices, for example, imaging versus flow, that you can make when you decide to use a small volume that maybe can make the test more accurate. I guess the other thing is that, there are, I'm sure, lots of different assays that our instrument just can't run because the number of molecules in a 10 microliter volume of blood is just not enough. There are all other things, for example, venous blood and capillary blood is not equivalent, right? There are chemical changes that happen when the blood goes from your, when the blood goes from your arteries through the capillaries into the vein. So we're particularly looking at things where the capillary and venous equivalence has already been demonstrated, but there are things where you just have to have a regular blood draw in order to use the current clinical diagnostics. Deep learning, I think, certainly has a role to play. We use it. I think deep learning is one of those technologies that probably is seen as a little bit of a black magic at the moment, right? That it can do anything. That's probably not exactly the case, but certainly it has a role to play. For example, trying to denoise or pull out very weak signals from a larger background, things like that.
Other questions?
So you show some very nice results. I guess the question is, what needs to be done before you can take these devices into the boonies? Because I mean, they look very nice, but I'm a bit skeptical about how long they would continue to work if they were out in the woods somewhere.
That's true. I would definitely agree with you. I mean, we are starting to do some of that stuff. So many of the data that we showed you today are things where we have taken the instrument outside of our laboratory and moved it someplace else. I wouldn't necessarily classify those locations as the boonies. They still have some amount of power and other things and not being run on batteries. I guess maybe like a different way to answer that question, because I don't think I have exactly a good answer, because we haven't done it, so we don't know exactly what are those challenges. But I guess what I would say is that another way to phrase that is that what's left to do here before you commercialize the product? Because in my view, those kind of very difficult questions are things that a larger team of people at a company probably need to solve. I was at a conference earlier this week, and I saw somebody who has made a very beautiful portable refractometer to do vision correction diagnostics in low resource settings. And they said that they agonized for months about how to design an eye cup that could work on many different kinds of people among many different ages and things like that. And all of those different, choices, I think, have to be addressed. Right now, our systems are basically stable as long as you're not throwing them around. But I guess in the real world setting, people are throwing them around, right? You can imagine a vet throwing something onto the back of his truck and driving over dirt roads someplace. And, I guess it's just the very detailed and mechanical engineering to realize that kind of robustness that we're not at yet.
I guess the follow-up question is who should do this? And, one of the differences is you have a $300 device, and I can see a company being really interested in selling a $300,000 device, but to build a $300 device and earn a profit on it is a bit more of a challenge, right?
That's right, especially because these are things that we're trying to design to be sort of consumer level cost, but not consumer level volume, right? So this is one of the things that we are struggling with. I don't think we exactly know what the answer is in terms of what kind of company is really going to commercialize this and what exactly are the markets. There are people, for example, this automated refractometer, where they're pursuing, NGO customers or governmental customers, things like that, where, people who are not necessarily in it for a profit motive see something. And then I think another thing is that there's a new class of companies that are starting to become more and more common, where they are not motivated by a 10x return, right? They're motivated by a 1x or 2x return, and they're interested in trying to maximize the social benefit as opposed to the sort of economic benefit of something. But I think all of these things are things that, if you ask who's going to do it, to be perfectly honest, it's probably not going to be me. I don't consider myself a particularly good businessman, but we hope that, you know, as we push the science and the engineering of this stuff forward, that there will be companies who start to see the, you know, the benefit to working together with us on this.
Other questions? I do have a quick question. So, even for some of your data, you seem to be a bit outside the CLIA certification, right? So what do you need to do?
Right. So the CLIA testing we did. So in some of the plots we show the testing compared with the clinical instrument. The clinical instrument or the clinical test is always better than CLIA. So even if we're a little bit outside that, we may not be outside the CLIA boundaries. And CLIA, for those who don't know, is the Clinical Laboratory Improvement Amendments. It's a criteria that says how good must a test be able to be run, or how good results must you be able to get from an untrained user in order for this test to be able to be operated outside of a certified laboratory environment. So here, these blue bars are the Clia limits, and you see we do have some points outside of that. The allowable percentage outside the Clia limits is 5%. So within it, but in some cases we're just within it, right? So I think that the things that we need to improve on are probably some of the data analysis, right? That I think the images that we get are totally fine. The sample preparation we're doing is totally fine. But again, we're doing these very, very simplistic, you know, almost ancient computer processing methods like template matching and other things. And if we go to something like a deep learning or like some other, you know, fancier method, we probably can improve this a little bit.
No other questions? No, thank you so much. Thank you very much.
Thank you.